And welcome everybody to the 2020 NABT poster session. I'm glad that everyone can make it. We've got an excellent slate of presenters ready to share their research with you. Before we get started, I wanted to take just a minute to thank the loads of people who made this year's poster session possible in this virtual format. It's certainly something new for NABT to try to do our poster session this way. And it took a lot of teamwork from a lot of people to make it happen. And so I just want to say thank you to NABT leadership and especially to the judges who worked diligently to look at the posters and the videos before our session and to assess each of those poster entries. I want to say thanks to those judges. This year they were Carrie Buckland, Valerie Haywood, Annika Metz, Anissa Lee, Aaron Baumgardner, Melissa Haswell, Jennifer Mraz-Craig, Jonathan Moore, and Rachel Pig. So certainly thanks to all of those judges and the work that they put in to make sure that our poster session could happen this year. I also want to thank all of the students who submitted their work this year. It's certainly super impressive that in the midst of a global pandemic that these students were able to carry on with their research and to persevere through certainly some trying times. And so thank you to all of the students and to their faculty mentors who were able to continue their work and to submit their posters to NABT this year. They put in a lot of hard work, and I'm certainly grateful for that. So to give everyone just a quick overview of how the poster session will work this year, we've got 3 categories in our student poster competition, and we'll go through those categories one by one, and then we'll have a chance at the end for a Q&A for all of the presenters. And so for the attendees out there, you'll notice in your Zoom, you have a Q&A button along the bottom, and that will be the place where you submit your questions to the presenters that we can circle back to during the Q&A session and give them an opportunity to address those. I would like to mention that when you submit questions to the Q&A, it would be super helpful for us if you could make sure that you specify to which presenter you're asking the question, since we will be addressing questions for all of the presenters at one time. So please make sure you just specify the presenter that you're asking the question to. So it'll make it easier for us to moderate those questions as they come in. The other thing that I would mention is please make sure you use the Q&A just for questions. Certainly some of you are going to have some very nice things to say to these presenters about the work that they are presenting, which is great. I encourage you to leave those notes for the presenters, but I would ask that you not do that in the Q&A here, just to make it easier for us to actually filter through these questions during the Q&A. What I would ask is for you to leave those encouraging notes and comments on their virtual chats in the actual virtual poster session itself. You're able to leave comments, or even if we don't get to your question during the Q&A, you can leave questions for the presenters on their virtual posters, and then they can check those and get back to you as well. So we will get started with our first category of posters this year, which are the mentored undergraduate research posters. The mentored undergraduate research category is an acknowledgement of the fact that mentoring students through research projects is a major part of teaching. And so we as biology teachers encourage students to take on research projects either in the field or in the lab. And this category is an opportunity for students who have completed lab or field-based research projects to present that to the NABT community. And so this year we've got 3 award winners in the mentored undergraduate research category. And the first one that we have, our 3rd place winner, is Bailey Edwards from Arizona State University. And she will be presenting her work on undergraduate perceptions of bioethics topics. So feel free to take it away, Bailey.
Thank you. Hello, everyone. All right. So yes, my name is Bailey Edwards, and I am an undergraduate researcher in Dr. Sarah Brownell's Biology Education Research Lab at Arizona State University. I have been working closely with Dr. Brownell as well as Liz Barnes and a fellow undergrad student, Julie Roberts, to study the undergraduate perceptions of bioethics topics at ASU. So we know that bioethics is important for understanding the relationship between biology and society, but the experiences of undergraduate students in these classes has been very underexplored. So we know from a previous study that religious students have reported feeling uncomfortable learning about bioethics topics in these classes because they feel that their perspective would be undervalued. And we also know that bioethics topics can be controversial for all students, whether they're religious or not. So with these small bits of knowledge in mind, we wanted to research students' comfort levels when learning about controversial topics in these courses. And we specifically wanted to look at students' experiences when learning about abortion and gene editing, because these are topics that are often covered in bioethics classes and they are particularly controversial. So for this study, this was just a pilot study, and we wanted to explore the experiences and perceptions of students in bioethics courses in preparation for a larger study that we are doing this semester. So this pilot study occurred over the summer. And we had 4 goals during this pilot study. We wanted to learn about students' tendencies to support abortion and gene editing after instruction. We also wanted to learn about students' comfort levels when learning about these topics. And we were specifically interested to see if highly religious students were less comfortable learning about these topics than non-religious students were. And lastly, we wanted to learn about why some students were specifically uncomfortable learning about these topics. And we were interested in looking at instructional methods that could increase these comfort levels in the classroom. So we distributed a survey for this study, and on the survey, we asked a variety of questions. Firstly, we asked students whether they supported abortion and gene editing. And for this question, students were given 3 options that they could select. They could state that they supported these topics in all situations, no situations, or only certain situations. And if they chose the only certain situations option, they were provided a list of scenarios to choose from, and they were asked to check the scenarios in which they supported the topic. Students were also asked their comfort levels when learning about these topics, and their options ranged from extremely uncomfortable to completely comfortable, and they were asked to explain why they felt this comfort level when learning about either topic. And then we also obviously had the basic demographic questions, and we were specifically interested in looking at students' religious affiliations and their commitment to religion or their religiosity for this study. So our survey results are shown in the bottom half of this poster. On the left, we have the graphs about students' tendency to support each topic, and then on the right, we have information about their comfort when learning about these topics. So looking to the far left at the graph that shows support levels for abortion, you can see that most students chose that they supported abortion in all situations, but many students selected the certain situations option for this question. And looking specifically into those questions, the specific scenarios, many students said that they supported abortion in cases where the mother's life or health was at risk from the pregnancy, or if the pregnancy was a result of rape. They felt like these were acceptable situations for abortion to be implemented, but they did not support abortion in scenarios where the parents weren't married and didn't want to get married, or if the parents just didn't want the specific sex of the child. And then for gene editing, most students selected that they only supported gene editing in certain situations. And again, zooming into those scenarios, most students felt that gene editing was acceptable in scenarios where it was being used to cure life-threatening or debilitating diseases. The students were much more hesitant when it came to using gene editing to enhance physical attributes such as height, eye color, or strength. And then looking to the data on discomfort, we actually did not see a statistically significant difference in comfort levels between highly religious and non-religious students, but we did notice that both religious and non-religious students felt some degree of discomfort learning about these topics, especially with abortion, and they cited various reasons for why they felt this discomfort. Some students claimed that their upbringing made them uncomfortable learning about the topic because growing up, abortion wasn't considered an option to them, so learning about it as a valid option made them uncomfortable. Other students who didn't support abortion specifically worried that by sharing their opinion, they might harm students who had personal experiences with abortion. And then there were also students who had personal experience with abortion who feared that they would be judged for their experiences if they shared their opinions. Based off of all of this, looking to the instructional methods that students critiqued, most students said that any discomfort that they felt was really just due to the nature of the topics themselves, rather than anything that the professors did to cause that discomfort. But one student did say that readings on controversial topics like these might be better off as optional rather than mandatory, just to alleviate some unnecessary discomfort there. So ultimately, our results showed that topics like abortion are sensitive to all students, whether they're religious or not. And it's important for instructors to realize that these topics have to be approached in manners that are sensitive, not only to students that disagree with abortion, but also to students that have personal experiences with it. And we're hoping that in the study that we're doing right now, we'll be able to get more specific suggestions from students on instructional methods that could help them feel more comfortable. So looking to the limitations of this study, obviously it was only a pilot study, so we have been working out some of the kinks with our official study this semester, but it was a pretty small population size. We only had 71 students, and we also were only able to distribute our survey post-instruction. So in the fall 2020 study, we decided to use 5 bioethics classes rather than just 1, and we're doing a pre-post survey in order to try to learn about how students' opinions towards these topics shift over instruction instead of just analyzing what their support levels are after instruction. So we're hoping that this will give us even more informative information, and I'm hoping to be able to share those results next year with you guys. Thank you so much for your time, and unfortunately I won't be able to be at the Q&A session, so if you have any questions for me, feel free to visit my virtual poster and leave them there, and I will be sure to get back to you.
Thank you so much, Bailey. Just as a reminder to our attendees, if you came in a few minutes late, as the presenters are giving their presentations and you have questions, please feel free to put those in the Q&A that can be found along the bottom of your Zoom session. I should also note that as questions are coming in, if somebody has asked a question that you like, you can give it a thumbs up, you can like it. And questions that are liked more often sort of rise to the top. And that way we can make sure that the questions that are very popular or that a lot of people seem to be interested in will be the ones that we can get to during our Q&A session at the end. So we'll move on to our second place winner in the Mentored Undergraduate Research category. And that is Amanda Gregg from Northwestern Connecticut Community College. And she will be presenting her work on annotation of the GEM-G bacteriophage. Please take it away, Amanda.
Thank you. Let me share my screen. Okay, welcome to my poster presentation. Let's get started with some background information. So the rise in deaths caused by bacterial infections over the years reveals a disturbing reality that we as humans have made for ourselves. If nothing's done about it, antibiotic-resistant bacterial infections will become deadlier as antibiotics continue to lose their effectiveness. However, hope in fighting back lies with a rather unexpected microorganism. This microorganism is called a bacteriophage. As the name indicates, bacteriophages are a virus that target only bacteria. They are species specific with regards to their hosts and usually only infect a single bacterial species or specific strains. Antibiotics, on the other hand, target bacteria indiscriminately, which has the potential to do more harm than good. In the past, up until recently, there have also been instances where bacteriophage have been used to treat patients as a last resort, and the results of these cases have shown great promise as an effective alternative treatment to use in place of antibiotics. For this project, our work this year focused on annotating the genome of a bacteriophage called Gordoniaphage GemG, with the hopes of contributing to the scientific community and inspiring young minds through mentorship. So for this project, our work was done in two parts, our work from the spring semester and our work that was carried out this summer with participating high school students. Our project began during the spring semester during a molecular genetics class taught by Professor Sharon Gusky. I, along with 3 other amazing classmates from the lab portion of this class, had the opportunity to learn more about bacteriophage and the significance of studying its genome by participating in the CPHAGES project. Our goal during this time focused on annotating GemG's genome to identify the location of all of its genes and to identify their functions, which laid the foundation for our work the following summer. This was made possible by Professor Sharon Gusky, who also introduced and showed us how to use the bioinformatics software like DNA Master, HHPred, Starterator, Famerator, and Pecan, as well as additional programs used during the summer. We used DNA Master to auto-annotate the genome. However, auto-annotation isn't completely accurate, so we had to go back and annotate each of the 75 genes manually to ensure the accuracy of our findings, using the other programs mentioned to support or disprove the results generated from DNA Master for each gene. So for a little context, HHPred, or otherwise known as Remote Protein Homology Detection and Structure Prediction, is a server that we used in conjunction with DNA Master in determining what proteins each gene in the genome coded for. So to use this tool, the protein sequence of a gene in DNA Master was copied and inserted into the server, which would generate a list of results and their probabilities. The next bioinformatics tool we used was called Starterator, a program that lets students determine the best start sites of a gene from a list of start site calls that have been made for similar genes in other genomes. Famerator organized protein coding genes into families of related sequences using pairwise comparisons to generate a database of gene relationships. Lastly, Pecan, otherwise known as Phage Evidence Collection and Annotation Network, is another bioinformatics program we use to document the information we gathered about the genes of GemG's genome. So mentorship of the high school students took place during the summer, where our work was taken one step further to find promoter regions and conserve repeats within GemG's genome. Mentorship added an extra layer to the project as it allowed college students to share and teach what they had learned in the spring to participating high school students. High school students in this project were able to build skills, participate in the research experience, and gain insight into subjects such as bioinformatics, genetics, and biology, and were also exposed to a plethora of engaging and insightful material, as well as weekly discussions related to the project and about bacteriophage. So to search for promoters and repeats, each college student with their assigned high school students were given a section of GemG's genome to study. I and Renee Brinkman, along with our assigned high school students, were selected to annotate the second half of GemG's genome in search of promoters and repeats. We mentored our students on how to use DNA Master and Softberry's BPROM feature. So to find promoters, students begin by using DNA Master's promoter prediction feature to find potential promoters, and then compared these promoters using the promoters found from BPROM feature. Any potential promoter generated by DNA Master's prediction promoter with a probability of over 0.5 and any promoter found from BPROM with a probability of over 50% were documented in an Excel file for future reference. Renee and I also worked with our high school students to find significant conserved repeats in the second half of GemG's genome using the BLAST feature on the Phages DB website. So being able to map GemG's genome this spring and summer helped us to determine what is known. So we are able to direct our attention to study unknown areas of its genome for future research. In the second half of the genome, DNA Master called 100 promoters in total. However, BPROM could only validate only 5 promoter regions upstream of genes 38, 45, 48, 63, and 71, which code for an ssDNA binding protein, a helix-turn-helix DNA binding domain protein, and a DNA polymerase III sliding clamp and membrane protein, respectively. The function of the promoter region upstream of gene 63 was unknown. Conserved repeats were located by genes 28, 33, 34 between genes 66 and 67, 68 and 69. The significance of repeats in genes 20 and 34 were undetermined, but 2 repeats in gene 33 code for topoisomerase primase. The repeat before the 67th gene codes for helicase, and the repeat between genes 64 and 69 code for a replosome organizer of a bacillus phage. Another repeat before gene 67 codes for reductase, while the last repeat before gene 53 codes for exonuclease recombination protein. So despite the limitations of the project, the information we collected seems small, but the results of a single annotation project like this contribute to a larger pool of information. Organizations like the Howard Hughes Medical Institute and the Graham Hatfuls Group at the University of Pittsburgh have made it possible for both professionals and students to contribute to the development of life-changing technology, medical breakthroughs, and effective medicines to be made available in the near future. Thank you.
Thank you, Amanda, for that great presentation. And moving to our last presentation in the mentored undergraduate research category, I'm happy to introduce our first place winner in that category, Hannah Oliver from Oklahoma State University. And she will be sharing her research with us on characterization of teosinte branched 1 mutants in Soteria viridis. So whenever you are ready, Hannah.
Thank you so much. Okay. Good evening, everybody. My name is Hannah Oliver. I am a senior at Oklahoma State University studying biology with an option in teacher certification. I've been researching and preparing for my student teaching this past year at OSU. And under the mentorship of Dr. Engel, Dr. Hu, and Dr. Dowse of OSU faculty, I've conducted research which I have entitled The Characterization of Testosterone Branch 1 Mutants in Soteria viridis. So, why should we care about plants and crops? Well, globally, there has always been an increased demand for crops for food and biofuel purposes. And to increase crop production, researchers like myself are looking into plant breeding of specific traits as a solution. One trait that is being explored in plants is branching, and a gene responsible for branching is testosterone branch 1, TB1, which has been identified in grasses such as maize. At high levels of expression, TB1 represses lateral branching, or in other words, increases apical dominance in a plant. In previous studies of testosterone branch 1, TB1 mutants have shown to change the architecture of the grass, most popularly in maize plants. And TB1 mutants have shown in maize plants to increase branching. And so we were curious about the effects of TB1 mutants in other grasses, specifically Soteria viridis. We chose Soteria viridis because of its close evolutionary relationship to maize. And additionally, Soteria viridis has been recommended to be an effective genetic model for all types of grasses. And thus, we hypothesized that Soteria viridis TB1 mutants would show increased branching similar to TB1 mutants in maize. And alongside this research, I created a research-inspired middle school lesson plan following next generation science standards, which is located in the middle of the poster. To start off my research, we obtained 2 seed stocks of F2 CRISPR-made Soteria viridis TB1 mutants, and we categorized these mutants as mutant 4 and mutant 5. We planted 60 of each of these mutants and 6 wild-type Soteria viridis as a comparison group. And in our characterization, we collected both phenotypic and genetic data. Our phenotypic data tracked both the timing and structures of our plant traits. And the phenotypic data we collected were time to germination, time to heading, tiller number at heading, the height at flowering, and the primary branching time. And all of these traits were analyzed using the Wilcoxon rank test, which is a form of hypothesis testing. And as for our genetic data, we collected DNA from our plant tissues and examined these extractions through the nanodrop program and agarose gel electrophoresis. And using our isolated DNA, we created stock solutions that ran through additional PCR, additional gel testing, and selected sequencing. We analyzed all our phenotypic data between mutants as well as between all three groups. And based on the Wilcox and rank test, and looking at table 1, between the two mutant populations 4 and 5, there was no significance in any of the measured traits except days to heading. And when we compare the mutants 4 and 5 to the wild type group in figure 1, we found that there was a difference in the tiller count between these populations. The orange overlap in the mutant populations indicates that there is no significance in these values. But when we compare the mutants to the wild type outnumbered both mutant populations in tillers. And in our genetic results, specifically our selected sequencing, we discovered 2 homozygous samples in our mutant 4 population. And within these 2 homozygous samples, we identified a 16 base pair deletion mutation that led to an altered transcript and an early stop codon, which is pointed out in figure 3. It is exciting to find homozygous samples because due to their rarity and sterile breeding potential in the future. Our discovery results showed that there were very few differences between mutants 4 and 5 and between all 3 groups that we tested. We had not expected the tiller count in the mutants to be lower than the wild type, and rather, hypothetically, with TB1 removed, there should appear much more lateral branching in the mutants than the wild type. However, because the opposite outcome occurred, we failed to reject our original hypothesis. But in future studies, we propose an expanded study on the morphological traits, including inflorescence, branching number, and tiller outgrowth. And furthermore, we would like to create an F3 population and identify the exact effects of a deletion mutation we identified. And lastly, based on this entire research, my lesson plan that I developed follows Next Generation Standards Middle School Life Science 1. And the purpose of this entire lesson plan is to teach students how organism growth is influenced by both genetic and environmental factors. And thus that concludes my research. Thank you.
Thank you so much, Hannah. That was great. As we are transitioning to the biology education research category, again, just a quick reminder to the attendees, in case you missed it earlier. If you have any questions for the presenters, please put those into the Q&A. You can find the button for that along the bottom of the Zoom session. And if, as you're filtering through those questions, if you see questions that you like or that you have a similar question to, remember you can give them a thumbs up and vote them to the top of the list. And that way we'll make sure that we can get to questions that seem to be common or that a lot of people have. Again, once all of the presenters have talked about their work, we'll have one big Q&A session at the end for all of the presenters. So on to our category in biology education research. This category really underscores the fact that NABT supports evidence-based teaching practices. And so conducting research into the best teaching practices or ways that we can improve student learning outcomes is important to us as educators. And so the biology education research category offers students an opportunity to present their work that they've conducted on biology education. In this category, we divide it into graduate students and undergraduate students. And this year in the graduate student category, we have one presenter. The pool of individuals that submitted poster in the graduate student category was a little small this year. And so we are going to acknowledge a single winner in this category. And this year we would like to award that first place prize to Erica Nadil from Arizona State University. She will be presenting her work entitled Call on Me, Undergraduates' Perceptions of Voluntarily Asking Questions in Front of Large Enrollment Science Classes. So when you are ready, Erica, please take it away.
Awesome. Thank you so much, Troy. All right. So hi, everyone. Thank you so much for having me today. It's a pleasure. I wish I could see all of you in person, but this will do. So for those of you who don't know me, my name is Erica Nadil, and I'm a second-year PhD student at Arizona State University, working in Sarah Brownell's Biology Education Research Lab. So for the work I'm showcasing today, I worked under the mentorship of Dr. Katie Cooper, an assistant faculty member at ASU. And so I will be sharing on Call Me undergraduates' perceptions of voluntarily asking questions in front of large enrollment science courses. And so as many of us know as instructors, maybe even teaching assistants and learning assistants, that eliciting student participation is a common teaching practice that many of us engage in. However, when we actually went into the literature, we found that there was very little on student perceptions of this practice and what can actually discourage students from wanting to speak out in front of the whole class, including asking questions and even answering questions. And so from this literature, we also know that the male voice can oftentimes dominate the classroom, and that students who fear being judged by others in social settings may actually be less likely to speak up. But when we actually dove into the literature regarding this, we found that there was actually very little research pertaining to these demographic differences. And so to actually fill this gap in the literature, I want to point you to my two research questions here. So the first one being, to what extent and why do undergraduates perceive other students voluntarily asking questions is helpful to them in large enrollment science courses? So in other words, when undergraduates hear others asking questions, do they find it to be helpful, and why or why not? And our second research question to fill this gap is, how frequently do undergraduates report asking questions in their large enrollment science courses during a semester, and what can discourage students from wanting to ask questions in front of the whole large enrollment course? And so to do this, we actually conducted this study as part of a course-based undergraduate research experience, or as many of you probably know, a CURE. And so in this CURE, we actually had 19 undergraduate students at the University of Central Florida who worked collectively with us to guide this research. And so as part of a CURE, students actually get to develop the research questions with us, and they actually get to engage in the full process of developing our methods also. And then what we did was we actually surveyed students from an R1 institution, specifically 417 students, and we asked them about their experiences asking questions in front of the large enrollment science courses. Specifically, we asked students how often they engage in this practice and what can discourage them from wanting to engage in this practice themselves. And so using binomial logistic regression, we wanted to actually test whether there were demographic differences among different student populations. Okay, so what did we actually find? So I want to guide you up to my results here. And so to answer this first big question is, to what extent and why do undergraduates perceive other students voluntarily asking questions is helpful to them in large enrollment science courses? And so what we found is that over 90% of our students reported that they found it helpful when others asked questions to their instructor in front of everybody in their large enrollment science courses. And when we dove into the demographic differences, we actually found that first-generation college students, or students whose parents did not attend any college at all, were actually 5.1 times more likely to perceive that others asking questions in front of the whole class was helpful to them compared to their peers. We then asked, and I want to just bring you down to this next table here, we then wanted to know, okay, well, what are these reasons why students actually find it helpful? And so to do this, before we actually gave our closed-ended survey out to students, I actually did 50 interviews with students to find these themes and factors that emerged from students regarding why they find it helpful when others ask questions. And so what I did with those themes was then I then turned it into a closed-ended survey where students could simply select a checkbox. And so for this, what we found is that 96% of students found that others asking questions to them is helpful because those students too have the same questions sometimes. And when we looked at demographic differences, we found that students with high fear of negative evaluation or this worry that they would be judged in a social setting were actually more likely to select this reason as to why it's helpful when others ask questions. We then found that nearly 83% of our students found that other students' questions sometimes help them to clarify their thinking. We didn't find any demographic differences here. Our next reason that we found why students find it helpful is that students sometimes feel uncomfortable asking questions themselves. And when we looked at the demographic differences, we found that women and students with this high fear of negative evaluation or this worry about being judged were more likely to select this reason. We then found that nearly 73% of our students found that other students' questions sometimes give them a different way of thinking about the material. And we found that underrepresented students and students with higher GPAs were more likely to select this reason. And then lastly, why students find it helpful when others ask questions, we found that nearly 42% of students said that other students' questions can sometimes break up the lecture, and that can actually keep them engaged in class. All right, so now I want to bring you over to my next finding and my next big research question. So this was, on average, how often do students report asking questions in their large enrollment science courses during a semester, and what discourages students from asking questions? So just to remind you, we know that over 90% of students find it helpful when they can hear others ask questions in front of large enrollment science courses. But when we actually dove into how many students actually engage in this practice, we found that nearly half of students on average, report that they never ask questions to their instructor in front of large enrollment courses. And when we dove into the demographic differences, we found that women were 2.4 times more likely than men to report on average never asking a question to their instructor in front of others in their large enrollment science courses. So then we asked, well, what are the factors or what are some reasons that can discourage students from asking questions in their large enrollment science courses? And so we found that nearly 75% of students said that they feel anxious when they have to ask questions. And when we looked at demographic differences, we found that women, students who have been in college longer, so juniors and seniors, and students with high fear of negative evaluation were more likely to select this reason. We then found that about 66% of students said that they can actually just look up the answer to the question themselves, and we didn't find any significant differences there. We then found that about 64% of students said that they worry others will judge them if they were to ask a question. And we found that students with high fear of negative evaluation or this worry of being judged were more likely to select this reason. We then found about 59% of students said they have the option of asking questions to other students during the class. And especially this was the case because a lot of the classes are in an active learning style, so students can actually just turn to their neighbor and ask a question rather than asking to the instructor. We then found that about 55% of students said they actually don't know the material well enough to even ask a good question. And we saw that students with high fear of negative evaluation were more likely to select this factor compared to their peers. We then found about 53% of students said they can just ask their instructor outside of class. We then found that 36% of students said it could actually take away from others' class time, so they don't want to be the one taking away from other students' learning. We then found that about 23% of students said that another student will likely ask my question, so I can just sit back and listen. And then lastly, we found that about 22% of students said that they don't think they'll actually get a detailed enough answer to their question during class time because of limited class time. And we saw that students who have been in college longer were more likely to select this reason as to what discourages them from asking. So now we know as instructors, as teaching assistants, and as learning assistants potentially, that over 90% of students find it helpful and they can hear others ask questions. And when we looked at demographic differences, we found that our first generation college students are more likely to find it helpful when they hear others ask questions. We also found though that even though it's helpful to students to hear others asking questions, only about 50% of our students reported that they're likely to even ask a question in front of the whole large enrollment course. And when we looked at demographic differences, we found that the male voice, consistent with the literature, can sometimes dominate the classroom, and that women also have questions, but they're actually less likely to speak up and ask those questions. So in sum, our big takeaway here is that these instructor decisions or teaching assistant decisions that we make actually have significant impacts on our students and their perceptions of the large enrollment science classroom. So with that, I'll take any questions. Also, I can pop my email in the chat if folks want to chat with me more. But thank you so much for this opportunity. It's been a pleasure.
Great job, Erica. Thank you so much. Now we'll be moving into the last category of posters that we have, which is the biology education research presentations from our undergraduate students. We have 3 winners in this category, and we'll go through them in order. And then again, following these 3 presentations, we'll have time for a community-wide Q&A So let me remind everybody, if you have questions, please feel free to drop those in the Q&A. You can find the button for that along the bottom of the Zoom session. And again, it would be super helpful to make sure if you do have a question that you indicate who you're asking that question to, since we're going to be addressing all of the questions for all of the presenters in one session. And so in the biology education research category for undergraduate students, I'm very pleased to turn the floor over to our third place winner, Aiden Link from the University of Arkansas. And he will be presenting his work entitled Student Ideas About Gene Expression, Making Proteins versus Punnett Squares. The floor is yours, Aiden.
All right. Hello, everybody. Let me share my screen real quick. Okay. Yeah, so I am Aiden Link, and I am a student at the University of Arkansas. This work was done through 2 summers of REUs through the Ember Lab at the Rochester Institute of Technology under Dr. Dina Newman and Dr. Kate Wright. And I also have 2 co-presenters today that go ahead and introduce themselves.
Hi, my name is Eowyn Coakley, and I'm an undergraduate at San Jose State University in California studying biology and chemistry.
And I'm Corinne Mills. I am an undergraduate at Florida Southern College studying biochemistry and molecular biology.
All right, so Eowyn, go ahead and start us off.
Okay, so in molecular biology, it would be impossible to communicate ideas without using representations, as these processes discussed are not directly observable. So we hypothesize that biology learners encounter difficulties because they do not picture the same thing as experts when they hear the term gene expression, which is a foundational concept. Thus, our research question was, how do students of different levels and experts conceptualize gene expression in pictures and words? In this poster, we will show you that undergraduate biology students often conflate gene expression with Punnett squares and traits. We use student and faculty generated drawings as a tool to investigate how novices and experts think about molecular biology. And during the semi-structured interviews, participants were asked to draw and explain the concept of gene expression. We then designed and implemented an online survey to further investigate student ideas about Punnett squares and gene expression. So undergraduate biology students were asked in several different ways to describe the link between genotype and phenotype and to choose the best visual representation of gene expression. I'll hand it over to Aiden, and he's going to discuss figure 1.
All right, so figure 1, this is the results from our interviews with our biology faculty and our biology undergrads. So there was about 17 experts of our biology faculty and 21 novices. And the faculty is represented in blue here, and the novices are in green. So on that first figure 1A, this is the results of the images drawn when we asked what is gene expression. So about 80% of our faculty drew the box and line, the kind of classic that you can see in B of the gene as represented as those rectangles. And about 20% of our students did that as well. However, what was interesting is about half of our students drew the Punnett square or some kind of like trait, this kind of thing, usually the Punnett square, for what is gene expression. And none of our faculty did that. So then we also coded the words they used to describe gene expression. So our faculty always talked about the central dogma of biology using transcription and translation. And about half of our students did that as well. But 70% of our students describe gene expression using traits and characteristics. So kind of this picture shows a lot what we were talking about, where students were describing gene expression as things with curly hair versus straight hair. Here is detached earlobes versus not detached earlobes. So that was kind of what they were going with, and that they were really confident with that. But our faculty were always here, we're talking about DNA being transcribed and modified, making mRNA and translated to make a protein product. And we did a 2 proportion z-test to compare these columns, and we found them all to be significant. And then this information was then used to inform our survey that I will let Corinne go ahead and talk about.
We developed a survey, Aiden said, using this data, and we had 51 undergraduate biology students take it. This survey asked students to select an image that was most similar to their mental image when they thought of that term gene expression. The image options that students were given are shown under the x-axis on figure 2, and the sample contained similar numbers of beginner, intermediate, and advanced students. 43% of beginner students, which is represented by the blue, selected a Punnett square, while 33% of intermediate students, which is represented by the pink, selected that Punnett square, and only 6% of advanced students selected that Punnett square, is represented by yellow. The majority of advanced students selected either the image showing an operand or their central dogma, which was in alignment with the expert mental models from the interview data. On the survey, we also asked in 2 open-ended questions, which asked the students to describe first how DNA leads to phenotype, and second, what the relationship between genes and traits were. Responses from the students were coded and keywords were tracked. The keywords that the students used are shown in figure 2B. Again, the same color coding aligns with the intermediate, beginner, and advanced students. Based on the keywords students used, distinct patterns emerged. Beginner students did not use words associated with the central dogma, such as transcription and translation, while more advanced students did. Less experienced students, the intermediate students, and the beginner students also talked about control using that word, which this suggested a very deterministic viewpoint of genetics, while the more advanced students seemed to have a better understanding and used the word influence, which also showed that they had a more sophisticated model of gene expression. One limitation of this work was the small sample size. We do believe that we achieved a represented sample of participants and were able to replicate the findings from the interview data using the survey.
So results from this project are helping us to uncover student thought processes and mental models that have not yet been previously explored. We've discovered a surprising phenomenon that undergraduate biology students commonly define gene expression using Punnett squares and phenotypes, rather than the expected process of describing transcription and translation, more in keeping with the central dogma of molecular biology. So our findings speak to common struggles students have in molecular genetics, and based on this, we want to suggest that instructors help students develop clear mental models of gene expression and to correctly interpret a range of different visual representations of molecular genetic concepts. Thank you so much for your time, and we look forward to any questions you might have in the Q&A session afterwards.
Thank you so much for that presentation. Very good job. Our second place winner in the biology education research category for undergraduates goes to Sienna Sin from Davidson College, and she will be presenting her work on maintaining student engagement during an abrupt instructional transition, lessons learned from COVID-19. Sienna, whenever you're ready.
Thank you. Hi, I'm Sienna Sen, a junior at Davidson College, and I will be presenting my research that I performed alongside Dr. David Wessner titled Maintaining Student Engagement During an Abrupt Instructional Transition, Lessons Learned from COVID-19. Because of the COVID-19 pandemic, many colleges and universities announced that all classes would abruptly transition to a remote format. Thus, the COVID-19 pandemic resulted in the need for emergency remote teaching. And many instructors simply didn't have enough preparation, time, or training to develop well-designed courses. As a result, early studies indicate that students became less satisfied with their courses after the switch from in-person to remote instruction. However, previous studies evaluating the best traditional online instructional practices have emphasized the value in collaborative, active learning modalities, as well as the importance of including a variety of activities to support the needs of different learners and students. Using this information, we investigated the effectiveness of several instructional changes that were implemented in an undergraduate microbiology course at our institution during the spring 2020 semester. We hypothesized that these modifications could help maintain student engagement with the course. Specifically, the changes that were made to the course structure to accommodate the abrupt switch to remote learning included First, a switch to a thematic focus, as the content for the second half of the semester focused exclusively on SARS-CoV-2 and COVID-19. Second, the use of pre-class introductory videos, which provided background information on the daily topics and articles that would be discussed. Third, the use of electronic forums, where students were invited, though not required, to share their thoughts on assigned readings and respond to the ideas of their peers. And lastly, synchronous classes were held via Zoom. After the semester, student attitudes and perceptions were assessed by means of a standard course feedback instrument that included several questions related to the specific interventions that were implemented when the institution announced that all instruction would transition to a remote format. Students responded to these questions using a Likert-type scale, and they could also provide open-ended responses. When looking at our graphs and selected student comments, our results demonstrate that first, all of respondents agreed or strongly agreed that the switch to a thematic focus enhanced the course, with many respondents citing the applicability and relevance of the material, meaning that the thematic focus provided students with a contextualized understanding of the content. This finding may be particularly important given previous research has found that connections to real-world experiences may increase student engagement and encourage underrepresented minorities to pursue STEM programs. Second, a majority of respondents agreed or strongly agreed that the pre-class introductory videos were useful. These short introductory videos help students prepare better for class. Third, most respondents agreed or strongly agreed that the forums were useful. Electronic forums allow students to engage more deeply with the material and see different perspectives. Fourth, all respondents agreed or strongly agreed that discussions during synchronous classes were informative. Together, these findings may suggest the importance of providing a variety of content delivery methods and platforms, and may reveal the true value of building an online learning community. It is important to acknowledge the general disadvantages of remote instruction, as well as the limitations of our particular study. Technological limitations prevented some students from fully participating in all class activities. Moreover, remote learning lacked the face-to-face communication that exists in traditional in-person instruction, as demonstrated by some respondents mentioning the challenges in replicating in-class discussions. Additionally, because our instructional interventions were implemented abruptly, we only were able to assess student attitudes about the curricular changes at the end of the semester. Thus, a pre-post change comparison was not possible. Overall, our results show that relatively minor instructional interventions can help students remain interested in and engage with course material, as we found that the thematic focus and specific instructional changes enhance the course during the switch from in-person to remote instruction. A thematic focus provided students with a contextualized understanding of the content. Short introductory videos help students prepare better for class, and electronic forums allowed them to engage more deeply with the material and created a broader learning community. We propose that our results suggest that similar, simple changes may also be useful in face-to-face learning environments. All of these elements can easily be incorporated into any course and potentially help to improve the student experience. Thank you so much for your time today, and I look forward to answering your questions.
Thank you so much, Sienna. Very nice job. And our last presentation for today is our first place winner in the Biology Education Research category for undergraduate students. And this year, we're happy to award that prize to Anna Abraham from Arizona State University. She will be presenting her work entitled, "Where Do Instructors Come From? An Analysis of Influential Institutions on Current and Future Faculty." Anna, whenever you're ready.
Thank you so much. All right, let me go ahead and share my screen. Okay. Thank you so much for the opportunity to be here today. I am so excited to share this project with you all. As Troy said, my name is Anna Abraham. I am an undergraduate researcher in Dr. Sarah Brownell's Biology Education Research Lab at Arizona State University. And for this project, I worked closely with my mentor, Dr. Dan Grunspan, who is now an assistant professor at the University of Guelph. So a little bit of background on this project. We are studying the professionalization of faculty. So that is, where did faculty earn their undergraduate degrees and where did they go on to earn their graduate degrees? Previous work has shown us that instructors are earning their graduate degrees from a small set of elite institutions, but it's unclear whether this pattern also exists at the undergraduate level. And we're interested in this because we think it really could be important for leveraging pedagogical change. Evidence shows that faculty are drawing upon their past experiences when they make decisions about how they're going to teach as professors. So in that case, we really should care about these past experiences and where they're occurring. We decided to study physics for this project because it's a smaller field than the life sciences. However, we would expect similar trends across the STEM disciplines. And we are also interested in exploring this in other disciplines in the future. So to go about this, we identified all the institutions that granted a PhD in physics in 2018. We then found all their tenure track professors in physics, and we searched for their undergraduate and their graduate degrees. And then we used this new list of schools to identify any institutions we hadn't yet looked at, and we repeated this process for a total of 6 rounds. We ultimately looked at over 7,500 faculty, and one of the unexpected and very interesting findings was that non-US institutions are training a large number of faculty. So looking at figures B and C, we can see where these faculty are coming from. So in figure B, we see that 431 of our faculty earned their undergraduate degree in China. And then in figure C, our top contributor was Germany, with 262 of our faculty earning their PhDs in Germany. And tables 1 and 2 kind of help put this into perspective. So these are the most common non-US granting institutions, non-US undergraduate granting institutions, excuse me. And we are comparing them to the US equivalent in terms of faculty production. So for example, Moscow Institute of Physics and Technology is responsible for training about the same number of faculty as Cornell University here in the US. And table 2 is the same most common non-US PhD granting institutions, and then compared to the US equivalent in terms of faculty production. So moving over to figure D, it's important to remember that we are interested in this because experiences at the undergraduate and graduate levels do impact how professors teach in the future. So we see that around 40% of our faculty are earning at least 1 degree outside the US. And initially, we thought that changing teaching practices at influential US institutions would create these large downstream impacts, but we can see that with the influence of the non-US institutions, we really should consider more of an open system and think about the influence that those schools are having. Another interesting finding is this imbalance in faculty production. So in figure E, this dotted line on the graph represents perfect equality. That would be if all of the institutions in our data set were responsible for training exactly the same number of faculty. However, we actually see this large inequality curve at both the undergraduate and the graduate levels. So we're actually seeing that 20% of institutions are producing somewhere between 70 and 80% of the faculty in our data set. And then in figure F, we are looking at the network of universities connected by where physics faculty currently work and where those faculty earned their undergraduate degrees. So this is this network here, but it's a little complex. So to kind of simplify this large network, we used a clustering algorithm to create this block model. And this grouped universities together based on similarities in how they are connected to each other. So there are a lot of interesting things here, but we really want to highlight the importance of block 4. So we can see block 4 contains 6 of the most elite US programs. And the arrows that connect block 4 to the other blocks, as well as to itself, are pretty thick, indicating that block 4 is responsible for training a large number of faculty, sending those faculty out to the other blocks, as well as to itself. And then figure G is the same pattern here with slightly different set of universities at the PhD level. So in this case, block 4 actually contains 12 of the most elite US programs. And again, we see these pretty thick arrows connecting to other blocks as well as to itself. So this imbalance in where faculty are earning their undergraduate degrees in addition to their graduate degrees is really important because these are the schools that faculty are earning their first experiences of a college classroom in. So knowing this information can give us ideas about what the most influential universities are. And then in terms of pedagogical change, we can think about maybe changing practices at a smaller set of schools will have these really large downstream impacts because of how much faculty they train. And then our finding that a large number of faculty are actually earning degrees outside the US also leads to further questions about how those faculty integrate their cultural backgrounds into the US classroom, as well as what this means for pedagogical change if we're thinking of an open system instead of a closed one, and what kind of interventions are actually going to be effective. So these findings are really what we're focusing on as we move forward with this project. Thank you so much to everyone for being here. I want to say a special thank you to all the members of the Biology Education Research Lab, to my mentors, Sarah Brownell and Dan Grunspan, and I'll be happy to take any questions during the Q&A session.
Thank you so much, Hannah. Very nice job. So now we'll be moving into our Q&A session. As we're going through this, please feel free to continue to drop your questions in the Q&A link. As a quick note, our first presenter, Bailey Edwards from Arizona State University, was unable to stay around for the Q&A session. And I do see a few questions directed her way in the Q&A. So for individuals that asked Bailey a question, what I would do is encourage you to go visit her virtual poster in the virtual poster lounge. And you can drop those questions directly to her in the chat for her poster, since she can't be here to answer. And then she will check that virtual chat with her poster and get back to you. So we'll move into a few questions for our other mentored undergraduate research presenters. So a few questions for Amanda. One is, have you considered making a manual or set of directions to explain how to use these search engines for high school students?
I think that's a great idea. Originally, what we did for our project was the college students actually prepared presentations for the students to talk more about bacteriophage, what they are, the different types, and how they can be applied to what we're trying to do. But I like the idea of having the material available to them for future reference, just in case, because it's a lot of material to have to go through and just try to memorize like all in one sitting. And so being able to have that for future reference, they can go back and they can remember what to do or have some guidance without having to answer the same question, 10 times. But great answer, great question.
Sure. And one other one for you, Amanda. What was your biggest challenge in terms of bringing this research into a high school classroom?
Well, I think the challenge for this project was the main challenge was we found out that some of our high school students were using Chromebooks. And unfortunately, we ran into some technical difficulties. Not too big of a situation. It was easily remedied, but supposedly Chromebooks are not compatible with some of the programs like DNA Master. So it was just all a matter of scheduling times to meet with the high school students and using DNA Master along with them so we can generate results. And that way they're still involved in the research process and they're not missing out on anything.
Okay, thank you so much. And we've got a few questions here for Hannah as well. So Hannah, one question is, why do you think that the wild type expressed less of this particular trait?
Thank you for the question. Well, funny enough, we actually expected our wild type to express less, but the actual events that happened is that our wild type expressed more traits. So they had much more tillering than our mutant populations. And this might be because there is more than one type of branching trait in a grass. So it might be a branching gene unrelated to TB1, such as MAX or BRC. We are still currently looking as to why our wild type express much more than our mutants.
And another question for you, Hannah. How many days does it take to grow these enough in order to make observations? Is this something that can be done inside a classroom without using artificial lights?
So my research addresses Ceteria viridis plants, and my lesson plan addresses lima beans. So for a Ceteria viridis plant, it would take at least 7 weeks before the inflorescence can fully emerge, but it does germinate within 1 to 2 weeks. So you could do Ceteria viridis plants in the classroom. However, I use lima beans when creating my lesson plan because it has much more identifiable structures and it germinates in less time and takes less time to make those data collection points, about a month. So you could use either plant, and it can be done in the classroom. And since all plants do use light, you would need some sort of light source for your growing plants. I'm not sure how well they will grow in the dark. So it can be done either way.
All right, thank you so much. So we'll move on to some questions for our presenters in the biology, education, and research category. And we have a few for Erica. So Erica, COVID has forced us to use live chats while teaching in person and remotely to students at the same time. Would you predict, what would you predict or might have learned about students asking questions using those different modes?
Awesome. That's a great question. So just to be clear, I don't have any data on what's happening with COVID. This was all done pre-COVID and in person. But if we were to hypothesize, based on the literature, we're thinking that if students actually have time to articulate their thoughts and to come up with questions before, even if they're in a live chat, they might actually be able to do this, or this could be helpful for those students that might need, an extra few minutes to think about the material, or they want to articulate it in a certain way. I'm also thinking though, so we're hypothesizing that that could be helpful, but then we're also thinking that, right, students that have high anxiety or might have anxiety in social settings could still potentially be worried in those settings, especially if this is happening synchronously, especially if it's an online course that still has 300 students that can watch somebody be on the spot. So that could be anxiety provoking for sure. I also just want to acknowledge though that we are actually trying to propose using an anonymous live chat feature, so students still have an opportunity to share their voice within the classroom and they can hear others ask questions, but it would be actually anonymous. That way nobody feels like they're on the spot.
All right. And a few of our attendees are wondering, would you expect the same groups of students would still be reluctant to ask questions and to the same degree if the classes were smaller in size?
Sure, another great question. So again, for the purpose of this, I only explored large enrollment courses, but there is literature suggesting that because students can actually form community, not only with other students in small enrollment courses, but with their instructors, they actually are more willing to ask questions in those smaller settings. And because there's actually a lot of literature on this, especially in seminar courses where there's discussion, we actually wanted to focus on large enrollment courses because what we found is that a lot of studies are actually lumping together asking and answering questions under this broader term participation. So that's sort of why we looked at large enrollment, but I would suspect that students are probably more comfortable in the smaller enrollment courses, especially because there's more buy-in from the instructor that it's going to be a discussion-based course, right? So I think oftentimes when I've been in a seminar, and this is my personal experience, the instructor says, we want students to talk to each other. And in a small enrollment course, each student has an opportunity to share out. So I think it's also a very different dynamic because in a large enrollment course, instructors run into this issue of, well, I have 300 students and I can only let 10 students speak at a time. And this means that other students' voices might not actually be heard. So yes, I think the dynamic is different and we would see potentially different comfort levels of asking questions.
All right. And one more question for you, Erica.
Sure.
This one is, according to the attendee asking the question, that in my mind, there are two types of questions. You have this may be a stupid question, and then they ask something about what was taught or was in the textbook, versus a second type of question where they're asking about a real world application. And so did you analyze the data based on the type of questions that were asked?
Awesome, another really good question. So for the purpose of this study, we actually did not look at the type of questions students were asking because this was not an observational study. This was simply just us asking students to report based on their previous experiences. So in the future, I think we would definitely want to be able to observe different classrooms with different types of, instructor practices, right? Looking at a class that has an instructor have 10 students ask questions and comparing that to a class that has students ask no questions at all. But I will say that when I looked at some of the data from the 50 interviews before I did this study, was that there were actually students reporting out that they are fearful of asking a stupid question. And this was definitely a concern by a lot of our students. One thing that we are hypothesizing, though, that could potentially mitigate this is that maybe some instructor buy-in could be useful, right? So especially in science, right, we know that science can be chilly and marginalized groups might be fearful of speaking out and doing science because they're worried potentially about, failing or being put on the spot. And I think actually if we can use some buy-in, that sends a message that it's okay to ask questions, right? It's okay to not even know where to begin to ask questions, right? Because this is often what we face as scientists anyway. So I think those are some of our future directions, but definitely a good question. Thank you.
Great. Thank you so much, Erica.
Yeah, thank you.
And now moving on to a few questions for our undergraduate researchers in biology education research. We have a few for Aiden, Awen, and Corinne. So one question is, it's reassuring that advanced students start using central dogma eventually, but how do you think that Punnett square conflation with gene expression arises in the first place?
This is a wonderful question, and it's something that we asked ourselves when we started to see this result coming up because it was so curious. And then when we spoke to professors or faculty, they would say, oh yeah, my students do have that conflation. And something we were thinking about was when we teach like Punnett squares and Mendelian genetics and the pea plants, and you talk about like seeing the green or the wrinkly or the smooth or the yellow, somehow with students understanding like the expression, they take it literally like expression of traits, not gene expression. And so our hypothesis or thoughts are that there's something happening there when we talk about like gene expression versus seeing phenotypic traits. So it's somehow conflated within those lessons. And I don't know if Aiden wanted to speak more to that.
Yeah, it just something we really noticed is they really latch on to it. So like when we would ask gene expression, that would be something they're like really confident with. They're like, oh yeah, I know this, and then draw us a big Punnett square, draw, the blue and brown eyes. So that's something they're like very like I think maybe like an easier concept or something that's really interesting to students. So they like kind of hold that really hard. And then gene expression may be a little more difficult, a little more like harder to grasp. So, but they know they have those, what they think are like the basics, the foundations of the concept in Punnett square, and they kind of just stick with those.
And another question for this research team one question or one attendee thinks that central dogma, the teaching of that really needs to start in middle school and high school. So how do you think that with your research that you can reach these teachers so that we can start the process of fixing these misconceptions earlier in students?
Yeah, that is something that is this is kind of early on, so that's definitely something long term. We would love like suggestions on what we think would be the best way to get into the classrooms. We're thinking like publishing in journals that maybe teachers would be able to read and latch on to and be able to grow, but we would love more suggestions on how we think that would be the best way to get it out there.
All right, thank you so much. And we have a few questions for Sienna. Sienna, do you think that your results would be similar this term when instruction in many institutions has been remote delivery from the start rather than a transition midway during the semester?
Yes, of course. Thank you for the question. I think that if we were to replicate this study, well, there would be some similarities between the results for this term and that of this spring semester, and that the students would appreciate the thematic course and find the electronic forums and pre-class introductory videos useful. I also think that, as we're seeing in early research studies, one of the main reasons that student satisfaction decreased following the transition to remote learning was that it truly was an abrupt transition, with professors scrambling to redesign these courses within a matter of days. And so now I would expect student engagement to be higher, as instructors have had sufficient time to plan and to structure and integrate these active learning modalities into their courses. And also, students these days are more familiar with the online learning environment.
All right. And another question for you is, how do you think your results relate to the earlier presentation on students asking questions? Do you think that the electronic forums make students more likely to ask questions?
Yeah, I do think that the electronic forums would make students more likely to ask questions and share their perspectives. Specifically when relating to the earlier presentation about students being hesitant to participate in these large classroom settings, because this would hopefully reduce anxiety related to speaking in large groups, given that students would be able to have more time to formulate their thoughts and responses, which relates to Erica's comment from earlier.
Great, thank you so much. And for Anna, the question for you, were you able to stratify by institution type? I'm wondering if there were any patterns or connections between different Carnegie classifications, for example?
Yeah, thank you for that question. It was actually really interesting when we went to do the block modeling and we used the clustering algorithm. We didn't purposefully stratify by any kind of ranking. But the way it just worked out based on we used structural equivalence. So we looked at how similarly the universities were connected to other universities in the networks to create these clusters. And it actually just kind of worked out that the more elite and prestigious institutions that we would expect from something like, the Carnegie classifications or any other ranking systems ended up being put together in the same blocks, because they do both send out their faculty and hire faculty similarly to each other. So yeah, we didn't explicitly look for it, but it did end up showing that pattern when we used the clustering algorithm. Thank you for that question.
All right, great. Thank you so much. And as we're wrapping up, I'd just like to extend another heartfelt thank you to all of our presenters. It's, as I mentioned, super impressive that they were able to carry on and present this research in the midst of the pandemic that we're facing. They were also very patient with us at NABT as we're trying to figure out what this poster presentation this year would look like. So they were very patient and understanding as we tried to work through some of those details. And a thank you to the attendees and the great questions that they asked our presenters. I would remind the attendees that if you didn't get a chance to ask a question, please feel free to visit the virtual poster lounge. You can still drop questions into the virtual chats for each of these posters individually. And again, you can also leave them notes. You can exchange contact information if it's a topic that you're really interested in and want to be able to stay in touch with the presenters afterward. And again, a huge thank you to the judges and the other individuals that made this poster session possible. Thank you all for coming, and I hope you enjoy the rest of the conference today.